Welcome to Pathogens and Immunity Expert Exchange. I am Michael Letterman, and with me is Neil Greenspan. We are editors of Pathogens and Immunity, a journal developed to serve the needs of researchers. Our guest today is Dr. Anthony Fauci, until recently the director of the National Institute of Allergy and Infectious Diseases and director of the Laboratory of Immunoregulation at the National Institutes of Health. Tony Fauci grew up in Brooklyn, New York, where he played basketball for his high school and both stickball and punchball with his friends in the streets. He graduated from Holy Cross in Worcester and received his medical degree from Cornell University in New York City, where he served as intern, resident, and chief medical resident. He completed fellowship training at the National Institutes of Health and in 1984 became director of the NIAID. For decades, Dr. Fauci guided our national response to the HIV/AIDS pandemic, and importantly, was the principal architect of the President's Emergency Plan for AIDS Relief, known as PEPFAR, that has saved millions of lives throughout the developing world by assuring access to life-saving antiretroviral therapies. In recent years, he's been the voice of our national response to SARS, Ebola, and COVID, occasionally finding himself in the difficult position of having to clarify the remarkable statements of our former president. Throughout this time, Dr. Fauci has led a large basic and translational immunology research laboratory, the Laboratory of Immunoregulation, that has revealed a trove of insights about HIV pathogenesis and trained scores of scientists who are now national and international leaders. He has received numerous honors and awards, including the National Medal of Science and the Lasker Award. It's a real pleasure to have you with us, Dr. Fauci. Welcome to Pathogens and Immunity.
Thank you very much, Michael. It's great to be with you.
Neil, why don't you get us started?
The first question I wanted to ask is, what early influences steered you to medicine?
You know, I think it goes way back to my family background. My father was a pharmacist in a small pharmacy that he owned in the Bensonhurst section of Brooklyn. And that was a very long time ago. That was in the 1940s and 50s. And at that time, the neighborhood pharmacy was sort of like the hub of the neighborhood advice about medicine. And unlike the chain pharmacies of CVS and Walgreens of today, it was kind of the doc of the neighborhood, family counselor, neighborhood psychiatrist, and person who you would go to if you didn't have enough money to go to a physician. You could get free advice from the pharmacist. So the idea about service to others was something that I kind of grew up with from the time I was literally a child. And then that was fortified by my high school and college education, which was Jesuit training in a high school in Manhattan, Regis High School, and then Holy Cross College in Worcester, Massachusetts. The theme of those educational institutions was service to others. And I felt the best way to serve others in the context of having an affinity for science was to be a physician. So as soon as I got into high school, I realized that that's something I wanted to do. I also wanted to be very close to people, so I took a humanities course, and it wasn't the standard road to a medical school. It was, classics, Greek, Latin, philosophy, together with enough science to get me into medical school. And that's really one of the reasons why I wanted to become a physician, a combination of wanting to be with people, but also having an affinity for science.
That's great. I wish more of our students had an experience in the classics. You don't find much of that anymore in our schools.
Yeah, I know that.
Just mention as an aside that I'm not one of those people who is excessively enthusiastic about so-called STEM initiatives, because I think they're a little bit too narrow. And I think they actually don't work well for a lot of people like yourself, who just described how valuable it was to get educated in the humanities along with the science.
I think if you do excluding one for the other, you miss out on parts of your education that have been very important to me. My being steeped in the classics, I think, helped me after I became a physician to really feel very, very strongly about the problems in society, the problems in civilizations, the problems about disparity and things. All of that I think was helped by my background in the classics.
Um, so you went to medical school, you got your degree in medicine. Why do you decide to train in infectious diseases and have a research career in immunology? Why there were so many choices? Why those two?
And I think it really reflected, what my own personal needs are and things that attracted me. The things that attracted me about infectious diseases, and I'll talk about immunology in a moment, was, wanting to be able to have a target of my work. There are many diseases that we all are aware of that have multifaceted components to it, cardiovascular, autoimmunity, GI, and endocrinology. With infectious diseases, you had a target. It was an individual agent that was causing the disease. Usually, the disease is preventable, and usually it's entirely treatable. If not entirely treatable, certainly suppressible, as we've done so successfully with HIV. Also, I wanted a disease that was serious enough so that the odds, the stakes are high. Again, I'm not talking in any disparaging way about other subspecialties. I'm talking about what fits my personality. I want a challenge that you got to get it right or the consequences will be dire. And infectious disease is one of those disciplines. With regard to immunology, when I was in medical school, now remember, as most people know, I'm a pretty old guy now. So I graduated from medical school in 1966 and started my internship in 66. At that point, human immunology was in its incipient stages of appreciation. And it was a field that historically is closely related to infectious disease. So I was excited about the knowledge that was rapidly accumulating in human immunology. And since human immunology, is so closely connected with infectious disease, one depends on the other, evolutionarily and otherwise, that I decided I wanted to have a combined research endeavor as well as clinical in both infectious diseases and immunology. And you might know from my history, Michael, that in the beginning, when I finished my I had a combined fellowship in infectious disease and clinical immunology. So I'm board certified in internal medicine, in infectious diseases, and in clinical immunology and allergy. And for the first several years of my independence as an investigator, I was more heavily involved in autoimmune inflammatory diseases where I developed therapies for some of the vasculitides. And, credit to my mentor, Shelley Wolf, who put me on that project. It was a highly successful project that took diseases that were uniformly fatal and were able to induce, 90+% remissions. I did that for several years until HIV came along, and then I completely turned around the direction of my career to study exclusively HIV in its very, very early years, right from the summer of 1981.
Your decision at that to go into immunology and infectious disease was obviously prescient, clearly prescient. And in those days, and I remember them well because I'm only a bunch of years younger than you are, the ID docs were the best clinicians around, which is one of the reasons why a lot of us decided to do ID, but they all studied bugs and drugs.
Right.
There were precious few ID physicians who had made the decision to be immunologists. So
Right.
Good choice.
Well, I think it was retrospectively, it was a good move.
You bet.
Yeah. So are there any particularly consequential or unusual insights that you believe help to guide your clinical and research careers?
I'd have to jump ahead a few years from the end of my training, Neil, and it's something I just mentioned a moment ago. When I saw the first MMWR in June of 1981 of 5 young, otherwise healthy gay men from LA who presented with pneumocystis pneumonia, I was very curious about it, but I thought it was, some kind of drug they took that suppressed their immune system. Then the second MMWR in July of 1981, which now was 26, curiously and phenomenally, all young, otherwise healthy gay men who were presenting not only with pneumocystis, but with Kaposi sarcoma and with other opportunistic infections, who now were not only from LA, but from San Francisco and New York. That was a particularly transforming moment in my career and my life, because I said to myself, apropos of what we were talking about, the relationship between immunology and infectious disease, here was a brand new disease. And if you look historically, when was the last time we found a brand new disease? I mean, like, never. It was just one of those things that this is a historical moment. It's a brand new disease. We knew at the time, epidemiologically, that it had to be an infection, even though we didn't know what the etiologic agent was. We knew it was destroying the immune system and it was killing people. So for all the reasons that brought me into infectious disease, it checked every box in spades. But also, I was trained as an immunologist and an infectious disease. And here is an unknown infectious disease that's destroying the immune system and killing people. So it was like, oh my God, I have to study this disease. And it was really interesting, Neil, because when I made the decision to turn around the direction of my career and give my autoimmune vasculitis project to someone else that I had trained in the previous few years, my mentors thought I was making a mistake. And they were wise mentors. They said, you're foolish. Don't give up your day job. You have an accelerating career where the trajectory of your career is very steep. Why do you want to start doing this, which likely is going to go away and not be much consequence? And then obviously it became clear very soon, even to my mentors, who were wise enough after a while to say, I think you were right in making that change. But that was a transforming moment in my career, that second MMWR in July of 1981.
I got similar advice from some of the seniors in my institution when I decided to get involved in HIV research in late 1982. A bit later, but they said, this is going to go away. This is not going to be an important thing.
It just goes to show you.
You bet.
Yeah, I intended to ask what specifically you gained in terms of insight, in terms of your success with developing the treatments for the vasculitis. As I recall, when I first heard about you, it was in the context of Wegener's.
Right.
Wegener's granulomatosis?
Yeah, which is now granulomatosis with polyangiitis.
Okay.
That's what it's called now. Yeah. Yeah, I mean, actually, that was an interesting point in my career. I give a lot of credit to my mentor, Shelley Wolf, and the younger people in ID probably don't know or remember who he was, but he was really one of the real giants in infectious diseases, along with people like Bob Petersdorf and people like that. Shelley put me on a project where we made a decision to do something that was very bold. We had experience as infectious disease docs. So even though I was doing an autoimmune study, I still was the infectious disease consultant, along with my other colleagues at NIAID, for the other NIH institutes. And we would get called often to the National Cancer Institute, who were giving combined chemotherapy for lymphomas and Hodgkin's and lymphoblastic leukemias. And one of their combined therapy always included high-dose cyclophosphamide. So we got the idea, wouldn't it be interesting if we could use low-dose daily oral cyclophosphamide at a dose low enough to suppress the aberrant immunological response, but not enough to cause such leukopenia that you put them in danger of infection. So we monitored their white count and kept it within a safe range, but continued on a daily basis. And to our amazement, the remission rate went from 0 to 93% with these vasculitides. And it showed for the first time that you can use a drug that's used for cancer under lower doses for non-neoplastic diseases. So, it taught me that sometimes you got to take majestic leaps and take chances, and the risk benefit is high risk, but really, really high benefit.
I think back to the early days of the AIDS epidemic and how horrendous it was, and how all of our patients were dying in front of us, and our interventions might slow things down just a little bit. And I also, but not enough. And all of our patients, most of our patients were dying. And there was an outcry of outrage by activists all over the country who said we weren't doing enough and we weren't doing it right. And somehow you managed to establish a collaboration between AIDS activists and researchers, which was unusual, but very, very successful. How did you do it, and what were the challenges in doing it?
Okay, so let me try to give some succinct background of it for our listeners, because I think it's important. Because back early on in the early to mid 80s, the scientific community and the regulatory community. The scientific community is the NIH and people like you and I, and the regulatory community was the FDA. And we were doing things for decades, even before you and I were players in this game. And it was a very rigid approach towards clinical trial. There's inclusion criteria and exclusion criteria. There's no possibility of getting access to a drug outside of a clinical trial. And when it got to the FDA, it would take them several years to approve an intervention. Well, this was for diseases that it worked very well for. But HIV was different, because it was a different disease that this approach was ill-suited. And the activists knew it. And they were saying, A, we don't think you're doing enough. But more importantly, we want to be part of the discussion. We want a seat at the table so we can talk about the design of clinical trials. We want to talk about the rigidity of the regulatory process, and we want to talk about the fact that we need to put more resources. Well, the scientific community at the time said, understandably, but completely inappropriately now that you think about it, we know what's best for you. We are the scientists, we are the regulators. And when I say that now, it gives me chills, because how inappropriate that was. So that's when they said, okay, we're going to start being confrontative, disruptive, and iconoclastic until you start listening to us. And that's what they did. And since I was a public figure and was one of the few people back then who was very visible with HIV/AIDS, I became the target of their confrontative behavior. And when they started to attack me, I said to myself, let me listen to what they're saying, because these guys are in great pain, they're angry, and they're afraid. So I found out that when I put aside the theatrics and I put aside the iconoclastic behavior and listened to what they were saying was making perfect sense. And I said to myself, if I were in their shoes, I would be doing exactly what they were doing. And that's when I said, let's sit down and listen to you. And once I did that, I invited them in to sit down, which surprised the hell out of them. That's the first time any government official ever said, let's sit down and talk about it. And that began a relationship which didn't immediately become a love affair. It took weeks and months and years to gain their confidence. And now, fast forward 30 plus years, the activist community and the advocacy community is an integral part of everything we do, about the advisory committees, about the community programs. So we now listen to them and we're better off for it. But it took a leap to say, even though you're attacking me, I'm going to listen to you. And that, I think, was one of the best things that I've ever done, was to make that decision.
In the trenches, it was incredible to all of a sudden find ourselves in these committees, in these groups, in the AIDS clinical trials group, working directly with folks in the community who were committed, knowledgeable, thoughtful, and the collaboration was, I think, highly successful. At a great level, it contributed to the enormous success of combination antiretroviral therapies that now have rendered survival close to that of the general population.
Right.
And so we've got other challenges now, and the big challenge that the activist community and scientists are worrying about is the cure. Are you optimistic that there'll be a scalable and safe cure strategy at some point in the near future?
I mean, I'm by nature a cautious optimist. If I wasn't, we wouldn't have taken the chances we did over the many years of my career. I think it's a high bar and a tough situation. I mean, the idea that we keep hearing yet again another cure by a stem cell transplant, with all due respect, I don't need another one to tell me anything. You know, we've had enough. We know that you can do it, but the idea of doing an ablation and a stem cell transplant is not the way to go.
Right.
So the idea is, can we figure out a way to eradicate the virus? And if not, can we keep it under wraps without necessarily giving daily antiretroviral therapy? And to me, the closest thing thus far, which isn't a cure, is giving an injectable every several months.
Right.
Now, that's not a cure in the eradication sense, but boy, it's pretty close when you can give a medication just every few months and keep a virus very, very, very well suppressed. That doesn't mean at all that we should give up on trying to figure out a way how to make, either excise or silence any other reservoir viruses without giving an antiretroviral. I think we should pursue that and continue. If we're successful, wonderful. If we're not, there are other means to keep people suppressed with relatively little toxicity.
Is your reason for saying that stem cell transplant isn't the answer one of cost and scalability, or is it on some other basis?
Cost and scalability is important, Neil. But if I am a person living with HIV and I'm either taking one pill a day with little toxicity, or I opt to go for an injectable every several months, why would I want to take the chance of getting an ablation and a stem cell transplant where you have a mortality rate that is way, way greater than my one pill a day? Why would I want to do that?
Well, so is the injectable actually available now?
Yeah, yeah.
Oh,
And it also can be used for protection prevention.
Well, one of
highly
the
effective.
one of the exciting things that everybody is all excited about, myself included, is lenacapavir. That the study that was given in cisgender women, 100% protection. Wow. You don't
Incredible.
see that very often.
really just in the past couple months, though, right?
Yes, of course. Yeah.
The question I have relating to vaccines is, what are your thoughts about the prospects for developing a truly effective vaccine for prevention in the next 5 or 10 years? And do you think there's any hope in terms of the antibody approach versus the CTL-based approach?
Well, put it this way. I mean, obviously, we've had from the very beginning, when we did the first, beginnings of a vaccine trial back in 1987, 1988, when we had naively were given an envelope that we thought was going to do what most other pathogens do, respond with a neutralizing antibody to an envelope. Didn't happen. Fast forward a few decades of multiple failures, as it were. We don't like to use the word failures because you always learn something from a trial. but trials that did not give us the result that we needed. If you look at some of the research that is now coming out of laboratories like Bart Haynes and Dennis Burton and Ian Wilson and people like that, where they're really right now able to engage the germline B cells, and with multiple sequential immunizations, you can mimic the evolution of broadly neutralizing antibodies. It's really, really tough. And the latest stuff that has come out from those labs isn't there yet, but boy, it's going in the right direction. So although I think it's going to be difficult, I think the key word you mentioned, Neil, was in the next 5 or 10 years. It's not going to be next year. It's more likely going to be 10 years than it's going to be 5 years. But I think we have the best minds in a combination of immunology and retrovirology working on it right now. And I again, am cautiously optimistic that we'll get there.
I mean, I've followed that work a fair amount. And the question I have, I think I actually published a commentary some years ago about this, is can you really imagine scaling up a vaccine based on multiple different immunizations, on the scale of millions to tens of millions of people? Maybe it could be more focused, but I think it's going to be a major challenge, especially given the social context.
You're absolutely right, Neil. I think it is a major challenge, but is it technically, scientifically possible? I think so. I've always felt, once you conceptually prove something, then it becomes an engineering problem of how do you get it to be scalable. So if we can conceptually prove you can do it, it's kind of like taking the first big computer that fits in a big room and then turn it into something I have here on my cell phone. That's what I'm talking about. I mean, let's conceptually prove it first.
Well, and just as an aside, an interesting example of this sort of problem is you no doubt were among those who saw the article by Francis Collins about his sort of devastation over the lack of utilization of the medications that cure hepatitis C.
Right.
And we actually interviewed, as one of the people in the series, Ray Shanazi, who had a major role in developing those drugs. And there's really an example of what you said before about the potential to actually effectively treat, in this case, amazingly cure a viral infection.
Right.
Huge percentage, 95+% of patients who got the drugs. And yet it's being very much underutilized, at least as Francis Collins described it in his op-ed in the New York Times.
Well, he was absolutely correct. It's tragic. As you said, oh my goodness, we're actually curing an infectious disease that causes cancer that is incurable once the cancer occurs. Like, holy smokes. Wow.
The AIDS treatment strategies are fantastic. Most people recover their CD4 T cells and their functional immunity, but a minority remain compromised. Happens sometimes. And do you think there's a way to enhance immune competence in those people? We tried IL-2. It increased CD4 T cells, but they didn't do what we wanted them to do. Do you think that additional studies of other cytokines like IL-7 are warranted, or other strategies to bolster immune responses?
The answer is yes. But again, I say that cautiously, because what we've learned from the IL-2 studies, that when HIV comes along and punches out at the clonal level, antigen-specific clones, you could expand all you want, but if the clone isn't there, you're not expanding the right clone. So I think you can get some degree of reconstitution, but I'm not so sure how much. But that would not prevent me from still trying other approaches.
Have you given any thought to why it is that the CCR5 Delta 32 mutation is as prevalent as it is in large parts of Europe? And what is your assessment of the various studies that have tried to indicate that there's a selective advantage for that allele in perhaps some other infection?
That's not my field. I've just followed it. And, there has been, as you well know, proposals that somehow or other a disease centuries ago, smallpox or some related disease, in which the selection for those who had that mutation essentially kept those people alive by protecting them against the things that was killing them, and selected to have a rich population. For example, Northern European Caucasians, much more so over Southern African Blacks. So there had to be something in history where that allele was protective against something that was killing large swaths of the population. But like many others, I don't know what that disease is.
So COVID shows up, becomes a pandemic, and amazingly, an effective vaccine was developed incredibly rapidly. So what are the implications of that for us?
A lot of major implications and collateral implications. The major implications, the importance of investment in basic and clinical biomedical research, because if you look at the investments that was made in the mRNA platform technology, you go back and look at the first paper that modified the mRNA to allow it to avoid an inflammatory neutralization of it to be able to be used as a vaccine, was a 2005 paper in Immunity by Katie Carrico and Drew Weissman. So that was like 15 years ahead of time. And then if you look at Barney Graham's immunogen design of the S2P mutation that stabilized the spike protein in its pre-fusion form, that made it highly immunogenic, they were working on that, Michael, for decades for HIV, because Barney was working with Peter Kwan, who was trying to get the right cryo-EM conformation of the envelope, and he used that cryo-EM approach to get the right stable conformation of the spike protein, which he started off with mares, and then as soon as COVID came along, he immediately took SARS-CoV-2, and it took him like a few days to be able to show that and put a mutation. And so it tells us about the power of basic and clinical biomedical research. But the other thing it tells us is something very elementary. When the body shows you that it can make an adequate immune response that can clear the virus and protect you, then that is a roadmap to a vaccine. If you get infected with measles or infected with polio, even though there's a degree of morbidity and mortality, the overwhelming majority of people survive and are left with immunity that protects them against reinfection. And when you have a pathogen that doesn't change much, like measles and polio, you can use the body's natural immune response as a roadmap to developing a vaccine. That's exactly what happened with COVID. The body induces a very good immune response against SARS-CoV-2, which is the reason why the overwhelming majority of people recover from COVID, and they get the SARS-CoV-2, they eliminate it from the body, which gets us back to the question you asked a moment ago about HIV, which is the reason why it's so tough to make a vaccine against HIV, because the body's natural immune response to HIV is not adequate. There's no examples of spontaneous clearing of the virus. There's a lot of, elite controllers that control it, but they don't clear the virus. So if you take that conceptual proof that it could be done, and then you take Operation Warp Speed, where you say the federal government will make an investment of billions of dollars to de-risk the companies to go in and make a major investment in doing the clinical trials in collaboration with the NIH, with the clinical trial groups, the HVTNs and those others, to collaborate with them but to also guarantee the company will not lose any money. So what Operation Warp Speed did is not only did it finance and bankroll clinical trials of 30,000 people per trial, but it pre-manufactured the vaccine before we knew it even worked. So that as soon as the clinical trial showed that the vaccine was highly effective, bingo, we had tens of millions of doses of vaccine available to go. So it's got to be investment in science and bold investment in combination with the pharmaceutical companies.
Our
The reason I
citizens,
say that, we can do that with any disease.
sure, citizens need to recognize that, and that being prepared is really key. And this was an enormous success. And I guess the classic term would be semper paratus.
You bet.
Are you aware of any pathogens or circumstances clinically that might make mRNA vaccines less advantageous than maybe older technologies?
No, the only thing that is curious, Neil, that I think we need to see how it does with other pathogens, because right now, I mean, obviously, there's a lot of work trying to apply mRNA to flu and apply mRNA even to HIV. One of the things that is curious is the duration of the protection of an mRNA vaccine. Now, I don't know whether that's a peculiarity of a coronavirus or the peculiarity of the mRNA platform. But we know when we vaccinate somebody, the protection is not measured in decades. It's measured in months to year.
Right.
Yeah, that's the issue that we've got to work out.
So, with regard to that issue and something you mentioned earlier, how should we define herd immunity with respect to COVID? I mean, how do we think of it?
Well, I wrote a paper on that.
Uh-oh.
Uh-oh. That's okay. It was a simple paper. It was called that we cannot apply the standard criteria of herd immunity. It's not applicable with SARS-CoV-2. And the reason is, it's simple. I could synopsize the paper in 30 seconds. One is that herd immunity is dependent on an immune response that is durable, measured in decades to a lifetime, and a pathogen that does not change. So you have clear-cut herd immunity with measles. Why? The measles that I got infected with as a child, because I was born before the measles vaccine, is the same measles that's killing kids in the developing world today. Number two, if you get infected with measles or you get vaccinated with measles, the duration of protection minimally is decades and maximally is lifetime.
Yeah.
Those are the criteria that you need for herd immunity, because if you have a pathogen that keeps changing, like the multiple variants of SARS, and if you have a duration of immunity that's measured in months, the entire concept of herd immunity is no longer valid. That's the point.
This gets in then to the next question, which is, and I know you've been asked this by other interviewers, but I think it's important to address how precisely to think about the effectiveness of the mRNA vaccines in limiting transmission, because people often talk about it in absolute terms, that it either does or doesn't. And I want to know if you would agree. My take would be that even if it isn't 100% able to prevent transmission, that doesn't mean it's not benefiting us by reducing the probability of transmission or the scale of transmission.
Yeah. No, I'm with you on that, Neil, because it doesn't do it to the 90% or even 80 or 70%. It does have some impact on transmission, number one. However, that impact is short-lived. So if you start off with the ancestral strain that we made the vaccine against, in that trial, it likely had a reasonably good effect on transmission, not 93%, but reasonably good. What we learned from experience. And the people who criticize the scientists and criticize the public health officials say, oh, you told us it was going to protect. We made an assumption that that protection would remain at a high level, and it didn't. It was lower than we wanted to begin with, and it didn't stay very long. What stayed long was the protection against severe disease, leading to hospitalizations and death. That was reasonably durable, not measured in many years, but durable beyond a few months, whereas the protection against infection was lower than that against severe disease and was much less durable. But in answer to your question, Neil, that doesn't mean there was no protection against infection. If you could get a little bit of mileage out of that, it would be worth it. So long as you don't, now that we know what the results are, you don't say, well, it definitely is going to protect you against infection. It's not. I mean, I'm a classic example of that. I've been vaccinated 6 times with, the primary series followed by a bunch of boosts. I've been infected 3 times. So not only does vaccine, but hybrid immunity with vaccine plus infection didn't protect me against getting infected 3.5 weeks ago.
Right.
I had a very mild infection, but I still got infected. Now, given my age, if I didn't have hybrid immunity, I might have died from the infection.
Right.
So we're trying to prevent this with immunization and social efforts. What about masks? How effective are masks? Even if we wear them, well, if we wear them not well, how good are they in terms of protection?
Thank you for asking that question, because there's a lot of misinformation and disinformation. A properly fitted mask worn consistently of an N95 and to a lesser extent a KN95 clearly is protective. There are studies now that clearly take the fog away from do they protect or not. They clearly are protective. 100%? No, of course not. Because people wear masks and then every once in a while they take them off because they have to go eat on a plane. I mean, people say, well, I wore it for the entire time except when I was eating. Well, sorry about that.
What about in the case of mask mandates? Do you expect those are they worth trying to do? Because clearly it must be the case based on what you just said, and that was my own expectation, that they're effective if worn properly. When we're applying it on a mass scale, is that, at least in this social context in the US, is that going to work in the future?
I think the mood of our society, Neil, has said in a perfect world, if you had a society that didn't have that strain of independence, you can't tell me what I'm going to do with my life, that mask mandates, the way a number of other mandates that we do, would work, because it would get virtually everybody to wear a mask. I think the pushback against any mandate of anything thus far may turn out to be counterproductive, unfortunately.
Heartbreaking, isn't it? So we had this pandemic in 2020, and we might have another one in time. Are we prepared? Do we have a good system for sentinel surveillance in our country that will pick up something quickly and tell us who it is and what we might be able to do about it?
Well, sorry to say, Michael, I don't think we're as prepared as we could be, given the painful experience we've been through. I look upon pandemic preparedness and response in 2 buckets. One is a scientific bucket, and the other is the public health bucket. And I think if we continue to build on the high A+ success of our scientific preparedness and response, as proven by getting a highly effective vaccine in a record time beyond anyone's imagination of 11 months from the time the sequence was made public on a public database and the monoclonal antibodies and all those are things that were done. If we continue to do that, we're good. What we really need to do is we really need to improve our public health response, because the fractionation between the individual patient care, going into an ER, going into a clinic, and the public health that is sort of guided by central organizations like the CDC, the local groups are not obliged to report things to the CDC. The CDC doesn't have power to say, you've got to give us all this information in real time. We don't have complete electronic medical records the way they do in some countries, where in real time you could know exactly what's going on in the community. We've got to do much better in that. We've got to build up the local public health system so that we have good coordination between the central and the local, and we can respond in real time. We didn't do a very good job with that. The other thing that is a lesson learned, that I don't know how we're going to change this, because it's become sort of like, a landmark of our society, is the worst time you could ever have a pandemic is when you have a profound degree of divisiveness in society, where people make public health decisions based on political ideology. The idea that when Trump said the CDC is recommending masks, for me personally, I'm not going to wear one. So what that did was that immediately made masking a political issue, so that if you are supporting the president, you don't wear a mask, and if you're not supporting the president, you wear a mask. So that there were fights of people wearing masks or not wearing masks. That's a public health decision. Why should people be fighting about that? Because it had a political tinge to that. So as long as we have that, we'll look at the deaths and the hospitalizations. The red states and Republicans versus the blue states and Democrats, the deaths associated with COVID and the hospitalizations were very heavily weighted towards red states because they under-vaccinated versus blue states because they vaccinated more. That should never be. There should never be avoidable deaths because of a decision that's made because of a political ideology. That is so tragic.
I don't know if you saw this, but 2 years ago, there was an article in New England Journal about this concept of having a safe and effective vaccine, an initiative to have a safe and effective vaccine within 100 days for any new putative pandemic pathogen. And I have some reasons for wondering whether this is really conceivable. And I think you provided some basis for understanding why extrapolating from SARS-CoV-2 and the experience with the mRNA vaccines is not necessarily fair for all conceivable pathogens, because, as you alluded to, the work that had been done on HIV and even more closely SARS-1 in the earlier 2000s provided an exact roadmap for what mutation to make, for instance, in the spike protein to stable.
Right.
And so this group had this paper in New England Journal, and I think they were operating on the assumption that you could just sequence a virus and know which gene it was encoding a key antigen and stick it in a vector with mRNA and have a vaccine that's safe and effective. And I'm curious to know what you think of that.
Yeah, well, the answer is that's wishful thinking in some respects. But what I think they mean is that if you look at the prototype pathogen approach that Barney Graham and colleagues wrote about a couple of years ago, it's to take the 7 or 8 most likely viral families: alpha virus, punyavirus, coronavirus, filovirus, all the flaviviruses, etc., and to make a prototype vaccination and immunogen with that, put it in a phase 1 trial to show that it induces an appropriate immune response. And then if you get a virus from that family, to make the rapid adjustments immediately, and then use the phase 1 study that you've already done to jump right into an efficacy study. The only thing about the 100 days that is a little iffy, I think you could probably identify what you want to do, get it in a vector, and start a clinical trial. And in 100 days, have a vaccine that you probably, if you have enough cases, show is effective, at least in a phase 2 or phase 2B. I don't know whether you could do a 30,000 person phase 3 trial, but you may get some information from a phase 2 or phase 2B. The thing that is going to be tough is to have enough vaccine in 100 days to give to everybody, because that's a production issue. It took 300 days to do that with Operation Warp Speed. I'm not sure you're going to be able to do that in 100 days, but you could probably have in your hand a vaccine that you're pretty sure works. And then you could start distributing it as it comes off the factory, the doses.
My concern would be that it's basically an example of inductive reasoning. And in your experience at Holy Cross, you may have come across David Hume.
Yes.
And he pointed out the limitations of inductive inference. And I don't doubt it could work.
Right.
But I don't think we can be confident 100% of the time that we might not be surprised by some virus that's a member of a known family, but in some way has diverged in important respects.
Right. The thing you're betting on, Neil, is that if you take the prototype from that family and you make an immunogen, that immunogen would be similar enough in its conformation that if you pick the most immunogenic form of that immunogen, it would apply to whatever the particular outbreak immunogen is.
Right. And also that it doesn't have unfortunate cross reactions.
Exactly. Yeah.
Something like that. And then the linked question, and then it'll be Michael's turn, comes out of a podcast I heard with this very young but very impressive investigator at MIT named Kevin Esveld. And he was saying that because of the risks of doing experimental work on putative or likely pandemic potential viruses in particular, that we shouldn't do any experimental work on those kinds of pathogens. And it's sort of along the same lines as I just argued. I'm very skeptical that you can be confident you know what you're doing with a new virus, even if it's part of a known family, with no experimental work in animals.
Oh, I agree. I mean, I have to respectfully disagree with that approach. But people get swept away by gain of function, and they usually have no idea what they're talking about, what the definition of gain of function is. But
actually, it's not easy to define.
yes, well, they try to do that when they were trying to make it less confusing. Hopefully, they'll straighten it out a bit more now. But the operational definition some time ago was well accepted. They want to relook at it now, that's fine. But the issue about not doing any experiments in an arena to look at predicting what might evolve into an outbreak is foolhardy. That doesn't mean that you can willy-nilly go in and start manipulating pathogens by people who don't know what they're doing, who are not trained, in which the result doesn't give you any information that's helpful. But you've got to get a group of people with outside influence from the community, scientists in good faith, to say, what is the risk-benefit ratio of doing an experiment? And if the feeling is that the risk is too great, don't do it. But don't completely ban studying anything that have to do with an emerging infection. That would be wrong.
about was any experimental work, not just putative gain of function research,
Yeah, but any,
about the pathogenesis of a
right,
particular virus, which again, even a tiny mutation can shift how a virus deals with the host. And, you see this with SARS-CoV-2, even within a strain, the outcomes in different people and the nature of the relationship between virus and host is quite variable.
right.
It's not as straightforward as, oh, well, we know that family, so we can predict everything.
The short answer to your question is that we shouldn't just empirically shut off research of emerging infections. Forget the word gain of function, that confuses people. But to say all research on emerging infection is really not a good idea.
A growing part of our community, and some of this is related to political orientation, not all of it, that is reluctant to get immunized. Fear of immunization, fear of the side effects, and skepticism about the value of immunization. How should we, physicians, scientists, address vaccine hesitancy in our country and all over the world, in fact?
Well, it's a very difficult problem. And one of the things that we've learned is that vaccine hesitancy and not wanting to get vaccinated is a broad spectrum of people, from those who are inherently anti-vax, anti-science, and those who are hesitant because they need more information. So you're not going to change the mind of a completely recalcitrant person who, no matter what you say, is against it. And that person is probably more anti-science than it is anti-vax. But there are a substantial proportion of people who are hesitant that if we, in a good faith, transparent way, reach out to them and try to get them to understand the data and the science associated with it, and don't sugarcoat anything, talk in real terms that no intervention is 100% safe, but the risk benefit of vaccinations over decades and decades and decades has proved overwhelmingly in favor of the positive aspect of vaccines. We just need to keep talking about that and not condemning people who are hesitant. We should not tire of reaching out to them and trying to explain to them in pure, simple scientific terms and risk-benefit terms why it's important to get vaccinated.
One of the issues that I took note of during the COVID, particularly the early months of the COVID pandemic, was use of the word airborne in describing pathogens. As you no doubt remember, initially people were saying that SARS-CoV-2 was spread through droplets. And then I think over time, eventually it was accepted that it can also spread as aerosols. And there were some people who were not using the word airborne or didn't think the word airborne applied to droplets, which I find odd because droplets are suspended in air. I'm just wondering if you have any thoughts about whether that term should be used for both situations.
I think that if you look at the aerodynamic experts who are now getting involved, they're even saying that droplets themselves hang around much longer than we thought they would. And true aerosol obviously floats around for very extended periods of time. So I think that the use of the word airborne is probably going to avoid confusion, that anything that you get through the air, be it a droplet or a classic aerosol, is an airborne pathogen. I would think that sooner or later, if we incorporate that, it's going to be less confusing. I mean, the aerodynamics people absolutely insist that this idea that if you have a droplet, it drops in 3 feet is not so.
So I'm going to switch gears a little bit towards the reason why our journal, Pathogens and Immunity, exists. We were very concerned, and many people are concerned about the skyrocketing profits of the for-profit journals on which scientific progress is dependent. And there was a nice op-ed in The Guardian a couple of weeks ago by Abizadeh suggesting that universities start taking on the responsibility for sponsoring not-for-profit journals. You think that there's a role for universities to do it. Will Georgetown be doing something like this? Case Western Reserve is right now. That's pathogens and immunity. Is there a role for universities doing this?
I can't speak for Georgetown because that's not the area that I'm involved with at Georgetown. That would be for other people. But I believe that we really do need to get much more accessibility of journal information to people without the skyrocketing prices. And I know the publishing houses get very disturbed when you do that, sure. But if a young aspiring researcher doesn't have access to information because they don't have enough money, that's crazy. So we've really got to figure something out. If the universities are the answer, then let's go ahead and do it.
Well, Michael carefully formulated that question to get you to use terminology referring to young people. And so what I was going to ask next is brilliant, Michael, absolutely brilliant. What advice would you offer to a young person considering a career in biomedical research?
The fact that a young person is considering a career means they have an interest in it. What I can tell that young person, as someone who has been involved in biomedical research now in my 56th year, I was at the NIH for 54 years, that the gratification and the feeling of accomplishment that you can get, despite all the roadblocks and the stresses and the strains of getting funded and some of the anti-science, it overwhelmingly slants towards the positive. So if you're considering it, I think it's something you should pursue because it's amazing how much fun it could be and what a great feeling of gratification you can have from working in the arena of biomedical research.
On a related topic, physicians sometimes make a decision to go in to also have a biomedical research career, as you did. And it's great to have physicians involved in research because they see what's happening to patients. And folks who train in basic science, get a PhD, don't have that opportunity, don't get that perspective. So keeping physicians engaged in biomedical research, I think, is a real important priority. But do they need to go to get formal training in science? Does a physician who's thinking about a biomedical research career need to get a PhD as well?
The answer, I think, Michael, is no, but with some explanation. If you're a physician and you've done your training, a couple of years of intern residency, and you want to pursue a career in biomedical research, but you want to capitalize on your insight that you have as a physician, but you want to be sophisticated enough in the science, you can learn in a fellowship fundamental basic science if you get into a lab that is funded enough to give you the time to put into fundamental basic research and learning it. Because you don't want a physician only to be the person that's conducting the clinical trial. You want them to be able to design from a scientific standpoint the important question that needs to be asked from a basic and then ultimately a clinical science standpoint. I was very fortunate. I don't have a PhD. When I took a 3-year fellowship at the NIH, I went to a very basic scientific laboratory, and I knew that, and I got anxious. I said, oh my God, I'm going to lose my clinical creds here. But I didn't, and I ultimately got back to being very active clinically. But for those 3 years, I was in a very basic scientific laboratory, and I published, in the JEM and the JI and those journals. So I learned basic research. It isn't as intense as a 4-year or so PhD program, but it got me enough sophistication in basic research to at least understand it enough to be able to design a fundamentally sound basic and clinical research project.
The path that you took to your career obviously worked very well for you and very well for us. So, Tony, thank you so much for being with us. Thank you so much for sharing your insights and perspective on science, on the epidemics, on infectious disease and immunology.
Thank you, Michael. Thank you, Neil. It's great being with both of you. I appreciate it. Take care.